That's good.
Okay, you're on.
All right, so press the button to continue. Well, hello everybody, and welcome to one of the op-ed talks for the Summer Institutes in 2020. I'm Bruce Weir from the Biostatistics Department at the University of Washington, and it's my pleasure to introduce our speaker today, Ed Buckler from Cornell University. So Ed Buckler is a research geneticist with the USDA and an adjunct professor of plant breeding at Cornell. And he serves as the genomics lead for the Innovation Lab for Crop Improvement. Ed has done a lot of work in plant genetics over the years, studying evolution, methods to take advantage of natural diversity to improve crops and improve sustainability. Recently, he's been talking about the implications of plant breeding for human health and regarded broadly. And I'm looking forward to hearing what he has to say today. So, Ed, take it away.
Okay, great. Thanks very much, Bruce. A little over 2 decades ago, I was actually at the Summer Institute taking it myself, and a little while after that, I was an instructor there. So it's really fun to get back and see you all again. So, of course, right now, medicine and its contributions to trying to preserve health are really at the forefront of what we're thinking about across the globe. But I want to also argue that there's 2 other areas that are really key, and those are food systems and climate change, where I think biology can play global impacts on contributing to a better society and Earth. So I want to start out with where we are with feeding the planet. In certain regards, we've made some progress, but if you look at what business as usual is going to be for the global food supply over the next, through the rest of the century, we're going to get to nearly about 11 billion people across the earth, and a lot of those people are going to have very poor diets, either poor nutrition or too many empty calories, while there still will be several billion people with insufficient calories or protein. The vision I think a lot of us have, though, is we shift those curves and they start looking like this, where one, we really reduce the number of people with insufficient calories, and that's likely to come from more alternative proteins and adapting our productive row crops to climate change. While dealing with the issues on producing a nutritious food supply really have to do with producing accessible quality fruits, vegetables, and microbial foods. And if you look at climate change, of course, we really need to shift our energy needs, and the NRC report in 2018 highlight this, that we can get to a carbon negative economy by the end of the century, where first we shift those energy production systems, but then biology plays a really key role in producing net negative greenhouse gas emissions by carbon capture. So what our lab's about and what we're trying to advocate for is to really accelerate breeding to improve global health and the greenhouse gas balance. And what do we mean by breeding? Well, it's really the design and selection of organisms for human purposes. And so why are we focusing on plants? Well, if you think about that global nutrition aspect, a lot of the key to healthy diets, not too much empty calories and so on, do have to do with having a sustainable supply of fruits and vegetables. And if you start thinking about the questions about carbon sequestration, over 80% of the globe's biomass is essentially held by plants, with much of the rest of it being held by bacteria. So we can't really get serious about carbon sequestration if we don't have plants and bacteria playing a complete role in those systems. And so what has breeding already been able to do over the last 100 years using the knowledge of genetics? Well, the first thing I want to highlight that standard breeding approaches, which I kind of highlight on the left-hand side, is about making crosses, evaluating them with an appropriate experimental design, and providing good selection. And that process, in the case of maize, for example, in the US, has helped increase yield 8-fold over the 100 years of the 20th century. But what breeding has become is much more model-based, and it's a model of genotype to phenotype, where essentially, if you can create a mathematical model that relates genotype to phenotype, then essentially as fast as you can cross organisms, you can advance them. And so in the case of a species like maize, that's every 4 months. And essentially, advanced breeding now for about a dozen species is starting to use these style of approaches. But the quality of that mathematical model to relate genotype to phenotype really is at the crux of trying to make fast progress here. And the issue is that we need to move from more, or just focusing on those about a dozen species that are focused on food yield, to a much wider range of species if we're really going to take on these two other challenges. Everything from fermentation, fruits and vegetables, fuel and polymers, carbon capture, and assisted migration, which is essentially helping native species migrate to deal with climate change. If you start thinking about the number of species where these breeding technologies and genomic selection could be applied, you're starting to talk about thousands of species need to have accelerated breeding. So how do we take systems right now that are only working on about 10 species and start applying them to thousands? Some of that's just straight logistics. How do you get the information systems available and into the hands of a much wider range of breeders, and so allowing them to use these advanced technologies? And that's what USDA's Breeding Insight effort that's just started out is working on, is trying to get those types of tools into the hands of a wide range of breeders so they can help make more rapid decisions and apply these approaches to a much wider range of species. But the second issue is we need better models to allow us to go from genotype to phenotype. And if you're working in a major crop and you have a single population, a lot of our models actually are already quite good. We can predict with 30 to 90% accuracy, depending on the trait and depending on the population size. But if you take those same models and you apply them to outside the population, sometimes the models may maintain half their accuracy, but sometimes they'll just plummet to zero. We essentially can have no prediction accuracy outside of the population, because a lot of what we've modeled is really in the relationships between those individuals. If you go to a specialty species, you may not have anywhere near the training data sets to develop those models on, and the accuracies are going to be lower. And if we try to go between species, we essentially have no accuracy between them. So the question, I think, for the rest of biology, of where breeders really need help, is can we start adding in mechanism into these models so that, perhaps at the low end we could double accuracy or provide a lot more transferable information, so that all those breeders working with thousands of essentially specialty species can have more accurate models and make more rapid, more accurate decisions. And this is coming down very quickly. Essentially, we can now sequence a genome for $2,500 a genome. And the Earth Biogenome Project is going ahead and starting to sequence, hopefully, maybe 1 million more species over the next 10 years. But if you think about what the cost is to functionally parameterize a species, it can be anywhere from $5 to $100 billion. Just think about how much we're spending every day to understand the human genome. And so the question is, how can we transferability really is the key there, because we can't spend that much money on all these different species. And so NSF characterizes this problem as really sometimes the rules of life problem. And I would actually say, I think we know a lot of the basic rules of life right now. I think what we aren't doing a very good job on is really parameterizing the functions of life. And I think that's where we can have a lot more progress. And if you think about it from a deep evolutionary perspective, this is the phylogeny of life. And the little clade down here at the base, this is where flowering plants are. And although it's a little branch of life, and that's that 80% of all the biomass out there. And so the real question is, how do we learn from the rest of these species and apply knowledge there? Or the same for a wide range of other clades. How do we leverage the information from across all of evolution to make useful genotype to phenotype models? So essentially the hypothesis we're testing and working towards is, we think that essentially biophysics can really be parameterized across all of life. And so that means we should be able to use models for predicting protein structure, for predicting enzyme activity, for predicting enzymatic flux. And I think we're starting to see machine learning models coming up from various groups that are having good transferability from one clade to another. When it comes to chromatin structure, RNA expression, translation, of course, those rules are not the same across all of life. Bacteria is doesn't have the same set of transcription factors that maize does. But a lot of those are shared across eukaryotes. And so I think it is within our ability to start creating models that really work across all of eukaryotes. And there's all sorts of evidence that say transcription factor binding works, have shared motifs and shared things across all eukaryotes. Now, how does that grammar work together? That I think is still the open challenge of pulling those together and coming up with more unified models there. And of course, there are areas where we're not going to have as much transferability. And one, of course, is plant physiology, how carbon and nitrogen move around a maize plant is different from yeast. But there is a lot of commonality across large clades. For example, across land plants, they have a lot of commonalities there. And we really need to start working and trying to produce models that work across that larger group of species. And so we've been starting to build models like that, leverage models that other people are creating, and essentially creating a computational or computational frameworks where we can plug in different models at each of these levels. And so we can start trying to get more transferability out there. So instead of just having a genome with tens of millions of SNPs in it, we can actually have estimates of protein activities at 10,000 genes. And so where are we transferring these models? Well, I think I think there's enough evidence out there across the scientific community that evolution is shared. There's lots of aspects that are shared. When we can parameterize those across, I think needs to be really rigorously tested, and we pick out the models and the approaches that are better at doing that. So essentially, I have 3 requests for our scientific community. I think the first is to test your model in another species. And I think it's totally fine. If it turns out that it doesn't transfer, that's fine, but report it. Second, we've been working a lot more in training and testing models in multiple species at the same time. So we actually train across those. And there's it's given us more observations, more transferability. And I think there's a lot of opportunities in this as multiple model systems can be combined together to train models. And then I think we as a community also really need to think about how we share the model with the community and community standards. There won't just be one. Let's not also have 10,000 different ways of defining these models that make the predictions. And I'll highlight KIPI as one of those attempts to make a standard. And so 20th century success in breeding looked a lot like this: careful experimental design, high quality data collection, and then really good mechanisms for making decision support. And that really helped reduce overall global poverty and food production. The 21st century breeding success is going to look something like this, where we're really combining the knowledge across all of our communities together. And I challenge you and all of us to the question of how do we really unite the data coming from all of our different communities together in meaningful models that can help address some of these really large global problems. And these are the folks who've helped me really think about these problems, and I'll be happy to take some questions.
Thank you. That's very nice. A lot of food for thought. So do feel free just to unmute yourself and ask a question. Ed, when you was talking about using multiple species, what kind of species are you talking about?
So the models we've started building have been, we've made models with both Arabidopsis and maize and sorghum in them. So just those so far. But we're certainly starting to think about, how can we start putting some models together that have yeast in plants in the same model. And for a lot of these transcription factors, for example, a lot of that regulation, there's really good evidence that, well, I mean, we know that they're phylogenetically closely related, that we should be able to start training models across multiple species there. And we've also just picked up human genetic models for certain aspects of chromosome structure, intron splicing, and found that some of them actually have very good prediction accuracy in maize without even trying. But it's been interesting. Some fail totally, and some are transferable. And right now I can't tell you exactly what's making transferable models versus these non-transferable models.
We have a chat feature if anybody would like to type in a question, or you feel free to unmute and call it out. Ed, what was that in your second to last slide you had about sharing? Was it Kipoy? What was that?
Yeah, so that's a framework. Actually, I do have a slide on this. This is a framework that essentially we weren't involved in creating it, but essentially they've created a framework for taking genomic data and having a whole series of machine learning models that allow you to run predictions through. So it's designed for sequence style data. So, and then training and retesting and evaluating them in a regular fashion. And I think this is, we've seen, some things in this work have worked really well. Some things I think still have some rough edges on it. But I think that basic approach that we share models across genomics in a unified fashion allows us to really learn from one another and really over time create better models.
Yeah, this is a whole new way. It's a whole new ball game, isn't it? It's impressive. Anybody else with a question?
No, nobody has posted up any questions in the last 5 minutes, and we're out of time.
Okay, so I want to thank you all for attending. This talk has been recorded. It will be available. So it is public. Yes, this is it was a public site, and the recording will be publicly available. So thank you again, Ed. This is food for thought.
My pleasure. Thank you all very much.
Talk to you all later. Bye-bye.